When understanding the details of how both transcription and translation work, it is important to figure out the directionality of each component. This is important because, as we already know with both DNA and RNA, these strands of nucleic acid molecules are set in certain directions. If we think back to the base structure of a nucleic acid, we label the directionality of the molecules based on the carbons within the central sugar. So for the adenosine monophosphate nucleotide, we can number the carbons in the central pentose sugar as 1, 2, 3, 4, and 5. Looking at this entire structure now, we can call this the 5' end of the molecule and this the 3' end. If we were building a new nucleic acid chain, the only way we could add to this end would be to connect the 5' end of this nucleotide to the 3' end of another one, and we would do the opposite for the bottom. Notice that each nucleotide is facing the same direction. We can now look at the direction of this entire strand and say that this is the 5' end and this is the 3' end. Now, why is this important and how does it relate to both transcription and translation? When we look back at these processes, either DNA or messenger RNA is being read in a specific direction to create a new molecule based on the genetic information present. What you need to remember is that in both transcription and translation, the process of creating new messenger RNA or reading the messenger RNA moves in the 5' to 3' direction. So for transcription, as we can see here, the RNA polymerase enzyme moves along the DNA and continues to add nucleotides to the growing messenger RNA chain on the 3' end, connecting the 5' end of the next free-floating nucleotide. When we draw an arrow to show the movement of this process, we can see the new messenger RNA strand is made in the 5' to 3' direction. For translation, the ribosome moves along the messenger RNA strand in the 5' to 3' direction, which moves towards the 3' end as the polypeptide chain is growing. Again, drawing an arrow to represent this movement, this is what we get. Taking a look at the process of transcription, it is important to note that the process can only take place if RNA polymerase binds to the DNA in a location that is upstream from the gene, which allows for it to be coded and the messenger RNA to be created. The section of DNA that is upstream of a gene and allows for the RNA polymerase and other transcription factors to bind is called the promoter. Different molecules, again called transcription factors, can bind to the promoter region, which can either activate the RNA polymerase to bind to the DNA or prevent the RNA polymerase from binding to the DNA. So, this promoter region is the location where gene expression is controlled, based on what transcription factors are present. As an example, in eukaryotes, the binding of general transcription factors like transcription factor 2D, which recognizes and binds to the TATA box, a common sequence element within the promoter, acting as an anchor. This initial binding recruits other general transcription factors, forming the pre-initiation complex, which then attracts and positions RNA polymerase at the transcription start site, preparing the enzyme to unwind the DNA and start transcription. The precise binding of transcription factors ensures that transcription is started at the correct location and regulates the rate at which a gene is expressed. You do not need to know any specific example of this for the exam, you just need to know what a promoter region is and the general idea of how it functions with transcription factors and the role it plays in gene expression. Now, when we think about DNA being coded for something, we generally think about a gene that holds the code to create a protein. But while our DNA contains many of these genes, there are also many parts of the DNA that are not coded into proteins. And some of these non-coding sections have other important roles necessary for proper cell function. So let's talk about a few examples of what non-coding DNA sections do, keeping in mind that when we say non-coding, we are referring to not coding a protein specifically. First, at the ends of our chromosomes, we have sections of non-coding DNA called telomeres. These telomere sections act like the plastic tips found on your shoelaces, basically holding the ends of the DNA together, protecting the strands from degrading. Again, they are non-coding, so there are no genes here that are transcribed or translated into proteins. Next, we have sequences of DNA that help regulate gene expression, which include the promoter regions we just discussed, as well as enhancer and silencer regions that generally sit upstream from a gene. They help regulate the process of transcription and are not used to be translated into proteins. Next, we have sections of bases that code for the creation of transfer RNA and ribosomal RNA. These are coded with a different type of RNA polymerase compared to what we see in the regular transcription process to make messenger RNA. Finally, we have other sections of DNA that are within gene sequences but do not end up being coded into proteins. These are called introns, and we will discuss these more on the next slide. Eukaryotic cells are unique because they have organelles, which include the nucleus, and when the process of transcription is complete, the messenger RNA strand must exit the nucleus to attach to a ribosome within the cytoplasm for translation. But before it leaves the nucleus, this pre-messenger RNA strand is modified in a few ways to become the final messenger RNA version that will be used in translation. There are two specific ways that the RNA is modified. First, the pre-messenger RNA transcript undergoes RNA splicing, where non-coding sequences called introns are precisely removed by a spliceosome complex, and the remaining coding segments, called exons, are simultaneously spliced together to form the continuous coding strand. At the same time, stabilizing and signaling factors are added to both ends. A 5' cap is added to the beginning, which consists of a modified guanine nucleotide. This protects the strand from degradation and is required for ribosome bonding. And a 3' poly A tail, a long chain of adenine nucleotides, is added to the end, which further stabilizes the messenger RNA and facilitates its export from the nucleus to the cytoplasm. With these modifications in place, the messenger RNA is ready to leave the nucleus and participate in translation. The process of pre-messenger RNA splicing that we discussed on the last slide is really cool because it can work in alternative ways based on the presence of molecules that regulate the process. So for example, in one cell, based on the molecules present, it could be spliced with these specific exons, and in a different cell, with different regulating molecules, it could be spliced with this series of exons. When these final messenger RNA forms go through the process of translation, two different proteins will be created because of the coding differences found within the exons that were present. But recall that the specific section of pre-messenger RNA was coded from the same gene. So this single gene, based on the cell that it is in, can be expressed as different protein variants without ever changing any of the DNA. You do not need to know any specific examples of protein variants for the exam. You simply need to know what this process is and the significance of the variation that it can create. Now, looking at the process of translation, let's dive a bit deeper into how the process works, starting with how the process is initiated in eukaryotic cells. First, an initiator tRNA molecule with the anticodon UAC has a methionine amino acid attached to it. It then binds to the small ribosomal subunit, which produces a unique ternary complex, which is just a molecule assembly of 3 different components. This complex binds to the 5' end of the messenger RNA strand with help from the 5' cap, via an initiation complex that the cap recruits. Once bound, the ternary complex scans the messenger RNA sequence with the initiator tRNA searching for a match to the anticodon it is holding, which would be the codon AUG. Once AUG is found, the complex stops via the linking of hydrogen bonds between AUG and UAC. The large ribosome subunit then docks to the small subunit, placing the initiator tRNA that is holding the methionine at the P site. The next tRNA, with the appropriate anticodon, links up to the A site and the process of translation begins. Refer back to the SL video to see the animation of how the elongation process works, which immediately follows this initiation process. At the end of translation, the amino acid sequence, now a protein molecule, detaches from the ribosome. But before this protein is fully functional, it will undergo modifications. We call this process post-translational modification. Some basic things that can happen during this process is the protein can fold as bonds are created between amino acids in the chain. Some amino acids can be removed. 2 or more amino acid chains of different proteins can be linked together, and or it can become a conjugated protein with the addition of other non-protein molecules, like sugars to make glycoproteins, for example. One or all of these things can take place in order for the final functional protein to be ready to use. An example of post-translation modification that you need to know for the exam is the production of a functional insulin protein. Insulin is secreted from beta cells in the pancreas and is used to balance blood sugar. We talk more about that in other videos. But for right now, we need to know that in order for insulin to function properly, it must undergo multiple post-translational modifications. Translation of this protein occurs in the ribosome on the rough endoplasmic reticulum. It usually creates a polypeptide chain called pre-proinsulin, which consists of 110 total amino acids. This chain gets modified in the lumen of the rough ER and has a sequence of 24 amino acids, called the signal peptide, removed from the N-terminal. The result is a new molecule called proinsulin with a total of 86 amino acids. Proinsulin then folds to stabilize itself, creating 3 distinct disulfide bonds. More detail on those in another video. After that, now in the Golgi apparatus, a sequence of 33 amino acids is removed in the middle of the chain, leaving 2 separate chains that are held together by those disulfide bonds. We call these the alpha and beta chains, and together at this point there are 53 total amino acids between the 2 chains. In addition, 2 more amino acids are removed from the C-terminal end of the beta chain. This leaves the entire structure with 51 total amino acids, which is now the functional form of insulin and is secreted to help regulate blood glucose. For the exam, make sure you know this process and the steps for the post-translational modification of insulin. New proteins are being created in the body all the time. To be able to support the creation of these proteins, cells need amino acids to build them. So, where do these amino acids come from? You get a lot of material from the food you consume, and the body also synthesizes some amino acids. But those 2 things alone do not meet the requirements needed to run a complete organism. Instead, cells have a way to break down and recycle amino acids from old, damaged, or misfolded proteins. This task is primarily carried out by large, barrel-shaped protein complexes called proteasomes. These molecular machines specifically target proteins tagged with a small protein called ubiquitin for degradation. Once a protein enters the proteasome, it is hydrolyzed into short polypeptides, which are then further broken down into individual amino acids. This effectively recycles the amino acids, making them immediately available for the synthesis of new functional proteins, thus ensuring the cell proteome remains healthy and responsive to changing conditions.